So hi guys, my name is Lydia Morley and I am a PhD student right now in the Small Link Lab at Texas A&M University. And today I'm going to talk to you guys about some concepts and methods from our new paper, Turnover Importance, Operationalizing Data Diversity, Quantify the Generalism Continuum, written by myself, Benjamin Crane, Gary Krupnick, and Daniel Smalling. So just to give you guys a quick overview of what I'm going to be talking about today, first I'm going to give you a little background on the project, why we started this, how we started it. Then we'll talk a little bit about generalism in general, and then we'll talk about what is beta diversity and how do we use it to calculate turnover importance. And then finally, we'll do a demonstration of the turnover importance function using this GitHub link. The link is here. It'll also be pasted in the chat. And then I also have a QR code later on in the talk. So we really started working on this project interested in cataloging and understanding pollinator relationships among Native North American orchids. Native North American orchids are sort of atypical when it comes to global orchid ecology and evolution. Not many of them are pollinator specialists. They're obviously temperate, but they're not tropical. They're terrestrial, not epiphytic, and North America is one of the most speciation disadvantaged continents for orchids globally. But that doesn't mean that they don't experience factors that drive their evolution and ecology in interesting ways. And we were really interested in really trying to understand how pollinator generalism specifically has played a role in the diversification and divergence of Native North American orchids throughout time. But as I'm sure you can imagine, once we started this task, we got into this real puzzle, which I'm sure many of you have experienced, and that is trying to understand how we should classify generalism at all. What does it mean to be a generalist? And beyond orchid-pollinator relationships, interspecies interactions are central to ecology and evolution, from habitat creation to predator-prey relationships to mutualism to parasitism. These things form the foundation of ecological relationships and they drive evolutionary change. And many of these relationships across time and space and systems are in one way or another generalized. And the different impacts of generalism and specialism on evolutionary and ecological trajectories has been of interest to us for decades. And what also has been of interest for decades is actually how to classify what is a generalist and what is a specialist. And we can all I think we're all very familiar with this kind of traditional and perhaps deceivingly simple dichotomy between a specialist and a generalist. And that is that a specialist uses one kind of resource. So here we have this insect over here, only eating one kind of food, whereas a generalist uses many resources. So here we have an insect over here eating multiple kinds of food.
Lydia, sorry to interrupt. People are struggling a bit with the audio. Your mic
a bit with the audio. Let me try this. Is that better?
Is that better?
Can you guys hear me better?
I think that's a little bit better.
Okay, cool. Sorry guys, I'm just in a hot windowless office, so I just had to turn my fan down a little bit. So apologize for that. Okay. So just to recap, for those of you who could not hear me, it's difficult to classify what it means to be a generalist, but we see generalism across broad systems and aspects of ecology and evolution. So again, this traditional ideation of generalism that is just it's kind of this simple dichotomy where a specialist is a species that uses one kind of resource. So here we've got this insect that's only using one kind of food. A generalist uses many. So here we've got this insect that uses multiple kinds of food. And a different but similar delineation I'm sure we're all familiar with is that a specialist has a very narrow niche while a generalist has a very broad niche. But even these categorizations can be based on arbitrary or vague characterization. Of course, a species might be a generalist in one aspect of its niche and a specialist in another aspect of its niche, and vice versa. So researchers often disagree about the number and kind of niche dimensions that should qualify species as generalists, or even as what makes something more generalized than something else. And of course, we all recognize that generalism exists on a continuum with specialism, and perhaps no species is purely one or the other. And in order to embrace this continuum, researchers have really devised two kinds of ways to conceptualize generalism. So there are niche-specific and interactor-specific measures. Niche-specific measures address the extent to which species' entire niches are generalized or specialized. So these typically rely on weighted means or principal coordinates of many niche dimensions and can be used to quantify generalist tendencies across geographic and ecological space to give a single unified picture of generalism for an individual species. So this can include things like ecological niche breadth, number of food sources, number of pollinators, number of different habitat types, etc., all compiled into one single measure. Interactor-specific measures, on the other hand, describe the extent to which particular relationships, such as this insect and its food sources, are specialized or generalized. Most of these methods are descriptions of species diversity at the community level, where generalism of a focal species is measured with respect to the diversity or richness of its interactors, or even with use versus availability. So regardless of category, all of these measures depend on species richness, such as the number of pollinators that a plant has, to quantify generalism. That is, all of these methods rely on the traditional ideation that a species is more of a generalist when it has more interactors and less of a generalist when it has fewer. And that may or may not be true. We'll get into that. So in our attempt to really quantify degrees of generalism and find a good way to understand how orchids are being generalists, we found that all of the available metrics really ignore a critical feature of generalized relationships, and that is that they are fundamentally spatiotemporally heterogeneous. So I want to talk about this in the context of orchids. So first, don't even look at this picture, just do logic. We can deduce that an orchid species which is specialized to one pollinator, so an orchid that only has one pollinator, is going to interact with that same pollinator throughout its entire range. So there's going to be no spatial variation in the identity of that orchid's pollinator because it only has one. However, consider the next up example, which is an orchid with two pollinators. So orchid A and B each have two pollinators, and they could even be the exact same pollinators for this example. Orchid A interacts with both species throughout its range, while orchid B interacts with only one pollinator in some parts and the other pollinator in other parts. Further, orchid B is specialized to one pollinator in every part of its range. So locally, it kind of probably behaves more like a specialist, whereas orchid A is not specialized to a single pollinator in any part of its range. So how do we account for this quantitatively or even categorically? Orchid A might have a uniform distribution of pollinators, which is more similar to a specialist, but it probably doesn't behave like a specialist locally. Orchid B, on the other hand, probably behaves more like a specialist locally, but it has a spatially heterogeneous pollinator distribution, probably more similar to what we expect from other generalists. So all of these features are critical components of our understanding of how interspecies relationships shape ecological and evolutionary trajectories. But we have no way to really account for these differences using available metrics. So to overcome this difficulty, we cashed in on existing beta diversity frameworks to quantify this spatial variation in interspecies relationships that we think would manifest over space and time. So very briefly, we take occurrence records from focal and interactor species, we clean them, we spatially bin them, we generate community matrices composed of interactors across each focal species range, and then we use Andres Baselga's R-package beta part to calculate Sorensen family multi-site dissimilarity. From this, we derive turnover importance, or T, which I'll call it, which is just the difference between turnover and nestedness component of beta diversity divided by the total beta diversity score. And so this essentially just tells us the extent to which interactor turnover contributes more to a focal species overall beta diversity score, while giving us some independence from total beta diversity. It gives us a standardized measure that ranges from -1 to 1. So those are a lot of parts, and I want to unpack each of them individually so that we really have a good theoretical basis for why we're using T before we start talking about how to use it. So I think the first question is, how does beta diversity have anything to do with interspecies interactions? I think we usually think of biodiversity terms like alpha, beta, and gamma diversity within the context of an ecosystem, a habitat, or a landscape. And what beta diversity really wants to describe is just whether or not there is compositional variation between the local areas among a community. And we can consider these concepts in terms of interspecies relationships, where rather than just a simple community in space delineated by whatever boundaries you choose, the boundaries of that community in space are just the area where your focal species occurs. So in this way, gamma diversity would just be the total number of interactors that a focal species have. And so for our example, orchids would be our focal species and our interactors would be our pollinators. So the species that we care about, and then the interspecies relationship that we're trying to describe. Alpha diversity, on the other hand, would just be the total number of pollinators that an orchid has at a local site. And gamma diversity would just describe, and beta diversity would just describe the variation among local sites across that orchid's range. So some of you may be thinking, cool, but what really even is beta diversity? So I just want to do a quick primer on what is beta diversity and what are the specific family of computations of beta diversity classifications that we use for T so that we're all on the same page. So there are tons of ways to calculate beta diversity. There are so many. But at the heart of all measures, we're essentially reaching for the same goal. And we just want to describe the extent to which two sites or many sites are compositionally unique. Or in other words, we want to know the extent of change in community composition as first defined by Whitaker. So we have two choices. We can look at differences between each site in a community, or we can try to summarize change over that entire community with one measure. And the original beta diversity measure devised by Whitaker was, in fact, part of the latter, which is a multi-site. So I'm going to do a small example to delineate these, and we'll be using the Sorensen family of dissimilarity measures because that's what we ultimately use for T. So we can calculate Sorensen's dissimilarity for each pair of sites within this community, where this is our function b plus c over 2a plus b plus c, where a equals the species common to the number of species common to both sites, b equals the number of species unique to the first site, and c equals the number of species unique to the second site. So we can do this here. So we'll calculate beta diversity for sites 1 and 2. So site 1 has 2 species unique to it, whereas site 2 has 0 species unique to it. There are 1 species that is common between both of them, and then we add the numerator and the denominator back down to get a final beta diversity score of 0.5. If we look at sites 1 and 3, site 1 again has 2 species that are unique to it. Site 3 has 1 species that is unique to it. They share 1 species with each other. We add the numerator back down into this part of the numerator back into the denominator, and we get a beta diversity score of 0.6. We can do 2 and 3. Site 2 has 1 species that is unique to it, and site 3 has 2 species that are unique to it. They share 0 species in common. We add the numerator back down to the denominator, and we get a beta diversity score of 1. And this seems pretty intuitive, right? Beta diversity should be the highest when there is complete differentiation within a community. So they share no species in common. So we should expect that beta diversity is very high. And pairwise measures can tell us a lot of things. So for example, we can tell that based on these measures, sites 1 and 2 are probably the most similar to each other, while sites 2 and 3 are probably the most different from each other. But all of these separate measures don't necessarily tell us, on the whole, how to understand the extent of change within this community. So we can calculate that using multi-site beta diversity, which is just a variation of the Sorensen family pairwise beta diversity. So we have proxies for each of these components. So this minimum would just be the minimum number of unique species between each of your site pairs. This maximum is the maximum number of unique species between each of your site pairs. This is a proxy for total number of shared species in both sites. And then again, we just have the numerator back down into the denominator. So if we look at this, we've got here the minimum number of unique species for each site pair. We can just go back to our original calculations here, and we know the minimum number for 1 to 2 was 0. For 1 to 3, the minimum number was 1. And for 2 to 3, the minimum number was also 1. So we plug those in. Here we've got the maximum number of unique species for each site pair. So we have 2, 2, and 2. And then we also have here, so this SIST here, it is just 2 times the sum of the total number of species in each site minus the total number of unique species in the community itself. So site 1 has 3 species, site 2 has 1 species, site 3 has 2 species. We plug that in, we subtract that from 4, we multiply it by 2, and then we add it to what is our denominator. So we get a multiple site beta diversity of 0.67. So this tells us that this community on the whole has a beta diversity score of 0.67 in general. And for T, we use multi-site measures. So from here on out, we're just going to be talking about these features of multi-site beta diversity. So only multi-site. The next thing we have to do is partition nestedness and turnover. So you may have noticed that for some of these cases, the differentiation between sites is simply due to one site containing some, but not all, of the species of the other site. In other words, it's just richness loss, such as between sites 1 and 2. This phenomenon is referred to as nestedness, and we talk about this a lot. On the other hand, differentiation between sites can be caused by each site containing unique species, such as for 2 and 3, where site 3 doesn't have any species in common with site 2 at all. This phenomenon is referred to as compositional turnover. So for our purposes, we want to determine when beta diversity is caused by nestedness versus turnover, and we want to know how much. So luckily, we can simply just calculate the Simpson beta diversity index, which is effectively Sorensen beta diversity independent of differences in richness. So it's just only the turnover component of beta diversity. And we do that, and the formula is actually just the same thing that we just went through, minus the sets of maximums. So we just get rid of the maximums, and we get a Simpson beta diversity or a turnover component beta diversity of 0.33. Now, this doesn't mean that 33% of Sorensen beta diversity is attributable to turnover. It means that the turnover component is 0.33, so about 50% of total beta diversity is attributable to nestedness. Sorry, to turnover. And we can sort of see that in our example where these outlined cases are representing compositional turnover here. So we see species replacement and species replacement in these two examples between sites 2 and 3 and between sites 1 and 3. And then to determine the nestedness component, because the Simpson beta diversity is just like a subset of total beta diversity, we can actually just subtract Simpson from Sorensen. So we can subtract this top one from this middle one to give us what is the nestedness component. So how much of this variation in our community is due to nestedness. That's 0.34, which again is about 50% of total beta diversity. And we can see that just like we have about 2 cases of replacement in this community, we also have about 2 cases of richness loss. So here is when you share species and you contain some, but not all of the species from the other site. So site 3 contains some, but not all of the species from site 1, and site 2 contains some, but not all of the species from site 1 as well. So now that we know all of the parts, we can calculate turnover importance, which is just turnover minus nestedness over total beta diversity. And this will range from -1 to 1. And it basically gives us a picture of how much more important turnover in a community is versus nestedness. And again, for a community, we're talking about a community of interactors over the range of a focal species. And this is good because it doesn't scale on the total beta diversity value. So it's a standardized measure that always ranges from -1 to 1, no matter what your beta diversity value is. So when T is above 0, compositional turnover is more important. When T is below 0, nestedness is more important. So when we do this calculation for our little toy community here, we knew that total beta diversity was 0.67, The turnover component, or Simpson beta diversity, is 0.33, and the nestedness component was 0.34. We take 0.33 minus 0.34 divided by 0.67, and that gives us negative 0.015. So what this tells us is that turnover and nestedness are pretty equal in how they're contributing to this community, but nestedness is slightly more important. So we're slightly on the side of that. So when t equals 0, this means that there is complete spatial homogeneity. These values are identical to 0 total beta diversity. And in our data set, we found that species with t equals 0 were putatively true specialists. Like we talked about, where if you only have one species, it's impossible for there to be any variation in the occurrence of that species, right? There's just one. But T could theoretically occur anytime the focal relationship is completely spatially homogeneous. So if my community just consisted of these 3 species in every single part of my community, and my orchid was interacting with them the same in each part of this community. Now, we think this is pretty uncommon, and I'm probably not very biologically realistic, but it is theoretically possible. And in cases where you find this, local measures of generalism versus specialism can capture everything there is to know about a species and its interactors, because there literally would be no change over space, except for maybe differences in local community structure, which you would also have to explore. Okay, so now we know what beta diversity is, and we know how to calculate T. Let's go over an example of T's extremes using orchids and pollinators instead of just shapes, to get an idea of how differences in T might impact the evolution and ecology of the things that we're actually studying. So if we look at orchid A, it has a T-score of 1. This means that there is perfect compositional turnover. In other words, orchid A is likely to associate with unique and separate sets of species in different parts of its range. So it's likely to engage in frequent host switching. Gene flow between populations might be highly structured. As you can see between site 1 and site 2, there's no shared species to mediate gene flow between the sites among this orchid's range. Whereas orchid B, on the other hand, has perfect nestedness. That is, all of its interactors are a proper subset of the most diverse set. So site 2 just contains some, but not all, of the pollinators from site 1. And this also means that there is probably a core set of pollinators that this orchid really associates with, while having some flexibility in some other parts. And all of this can tell us some really important things. So of course, turnover importance simply just tells us the extent to which interactor beta diversity is caused more by turnover, like in option A, or nestedness, like for orchid B. But understanding these relationships and being able to have something that indicates the nature of the spatial relationship between the species that you care about can tell you about the evolutionary trajectories, the ecological stability, and the geographic range dynamics of the generalist species that you're actually interested in. So that's great, but does this thing that we think turnover importance tell us actually tell us anything unique, or could we just use other available measures? So we ran a bunch of simulations and we compared T to a ton of other available metrics for quantifying generalism. And I just want to share briefly, because a lot of these are just, boring graphs. And if you're really interested in it, we've got them all in the paper. So I just want to share what I think is one of the most important take homes, which is being able to answer, how do these other available metrics track changes in T? So in other words, is T capturing something unique, or can it be described using one of these other metrics? And so we can clearly see here that as T changes, other metrics don't really change predictably along with it. So they're poorly tracking changes in T. And the important thing here is that all of these measures that we tested require a single large community matrix, irrespective of space. So you can't separate occurrences by sites within the range of the species that you care about. So it homogenizes variation across the species range. They're highly correlated with overall species richness. And so they have trouble capturing that compositional variation in space or in time that we think turnover importance does capture. So that is to say that this shows that T is really capturing some feature of generalism that these other methods just don't. And of course, that is not to say that T is end all be all for methods of calculating generalism. It's a complex issue, and I think that it should be approached from many different angles. T fails to capture some features of generalism that these other metrics can capture. And that's namely use versus availability, abundance, and relative generalism within a community at local time-restricted scales. So we present T not as an alternative, but as a complement to these available metrics. So basically, use T if you want a single number that describes how interactor composition varies for a single species across space and time. And you can also use it comparatively if you want to understand how that variation has changed for, say, for example, within a phylogeny of closely related species. Use other metrics like D prime or S if you want to understand interspecies dynamics at the local level, referentially to the focal species constituent community in that specific place at that specific time. Okay, so we know what T is and when to use it and why we might want to use it. Now we're going to learn how to use it. So we're going to go through this example and detail all the moving parts because there are many. There are actually just 3, but it feels like a lot. So first, in order to calculate T for your system, you need 3 basic components. So SP1 or species 1, which is just a community matrix for the focal species. So in our example, orchids, where the rows are sites and the columns are species. You need SP2, which is a community matrix for interactor species. So for example, pollinators, where the rows are sites and the columns are species. You need SPREL or species relationships, which is information about the associations between your focal species and your interactor species, which is a long form data frame with the names species A, species B, and value. So now we're going to paste the link into the chat for the GitHub if you want to follow along this. Yeah, we're all pasting it. Everybody paste the link into the chat. If you want to follow along, this QR code will also take you to the GitHub page, but also feel free to just watch because we've got everything taken care of. You don't have to follow along with the real example, just make it. So I'm going to give everybody who wants to actually code along with us just a little minute to get to the website. I'll just impatiently sit in silence until I feel like you guys have had enough time. Okay, I think that's been enough time. Okay, so our raw data looks like this, and this is just the first 3 lines in the first 3 columns for our example data frames. And so it just so happens that for species 2, all of these species like don't actually occur in any of these sites, but that's of course not always the case. So naturally, really importantly, your sites for your species 1 and your species 2 should mean the same thing, and that is that they should refer to the same physical place. So for example, site 1 for species 1 should refer to the same physical location on Earth as site 1 for species 2, because we'll be combining these and using them to actually calculate T. So we've just got here for SP1 the orchids that we care about, these 3 orchids, Spiranthes verticillata, or sorry, Isotria verticillata, Spiranthes casei, and Spiranthes lacera, with the sites over which they occur. SP2 are the potential and actual pollinators of these orchids and the sites over which they occur. And then SPREL is just the data frame where we've got our focal species, our interactor species, and whether or not they actually have a relationship. And so for this, if you want to do this, you can have each interactor species be listed for every single focal species that you have on your list and just have a 0 if they don't have a relationship. Or as you'll see, the way that I actually tend to format it is I just only have single like 1 value. So Spiranthes casei and Andrena miranda don't actually interact. And so I just take that out of my initial data frame. The function takes care of that, but I just find that it's easier for when I do my later work with the species relationships that I just have a data frame that just only contains rows for like orchids and pollinators that do actually have a pollination relationship. So we can actually just run this really quick. If you're following along, go ahead and paste the stuff into your R, and you might need to download a bunch of packages. So sorry, but I'll show you what it looks like here. So there, I've just pasted the full function into a script in my R, and I get that function into my functions, which is called tcomp. I download my example data sets, and all of this is on the GitHub. So we can look at the example data sets. Here's SP1, SP2, just like we talked about, just community matrices. We have SPREL, and you can see where I've just gotten rid of all of the zeros. So my data frame only contains species that actually have relationships. And then we just run tcomp. And there we go. It's done. At the bottom here, you'll get, and it goes pretty fast. It depends on, of course, how many focal species you have in your system, but that took just a couple seconds. So it's not super computationally intensive, and it goes pretty fast. It'll output all of your species, all of your focal species, and the relative beta diversity metrics here into the console, just so that you know that it's finished, but also it will output them into a data frame. And so let's look at some of I want to talk about first the intermediate output. So if you're following along in R, you can type test, little arrow, core list 1 brackets data. And this is just the first community matrix in the list. So you also have all of these if you wanted to use them for later. The function saves all of these for you. So this is the actual data frame that R uses to calculate T, which is just all the sites are restricted to only occurring over the range of Spiranthes lacera, and all of the members of the community matrix are only those pollinators of Spiranthes lacera, which is just the first species in our focal species list. So thus, T gives us a measure of how pollinator composition changes across sites only within the range of pollinator of Spiranthes lacera. So this is what we actually use to calculate T. So it's a community matrix that is kind of an amalgamation of the 2 community matrices that you first import into R. So if we look at the final output, this is our data frame, which is just SPA beta. And so that is just all of the beta diversity measures for a focal species. SPA is all of your focal species, so species A. And then it will give you the 4 beta diversity measures that we've talked about today. So you get total SOAR, which is just a total Sorensen family beta diversity multi-site. You get Simpson and nested, which are the turnover and nested components respectively. And then you also get turnover importance, which is again, just the turnover component minus the nested component divided by the total beta diversity score. And we have that for each of our species that we care about. So if we look, Spiranthes lacera has a turnover importance of 0.942. So it's got a lot of, so compositional turnover is really important for it. Spiranthes casei, on the other hand, is a turnover importance score of -1. So nestedness is really important for them. And Isotria verticillata has a value of 0.1, meaning that there's pretty even distributions of turnover and nestedness across that community. So once we have our results, we can use them to form conclusions about how our focal species associate with interactors across space and how those interactors might mediate ecological and evolutionary interactions, such as migration, gene flow, and long-term survival in specific areas. So from the running example that we just did, these are the actual results of the actual distribution of pollinators for these orchids in space. So this top row is showing us richness gradients, so changes in pollinator richness, while this bottom row is showing us distinct compositional clusters of pollinators. And so we can use our distribution data sets that we've had to kind of create these and help us understand really what T looks like. So again, Spiranthes casei has a T value of -1. So if we look, all of the compositional differentiation, all of the compositional structuring to its pollinators across its range are identical to differences in richness. That is, that is just the definition of turnover importance being -1, but it also tells us the nestedness is really important for Spiranthes casei. So if we're interested in understanding which pollinator relationships are more important, we should probably try to understand which pollinator relationship exists in the least rich part of its range. But at the same time, if we're also interested in maybe trying to conserve certain populations, we should look at the areas where, of course, there's a lot of richness loss and it has fewer of its total possible pollinators. On the other hand, for something like Spiranthes lacera, where richness doesn't really tell you anything about pollinator compositional clusters, we might be more interested in trying to understand how gene flow is structured across this species. And so we might want to go out and do targeted community studies where we look at the, genetic differentiation between the Spiranthes lacera occurring in pollinator clusters A, B, or C. And then, of course, Isotria verticillata has an intermediate value, and I think that it really emphasizes the importance of having this continuous variable and understanding, again, that generalism is, of course, a continuum. And Isotria verticillata is going to be subject both to changes in richness, and it's also going to experience some compositional differentiation. And so we can't prescribe solely one or the other course of action for this species. But turnover importance can help us understand when these different phenomena are occurring and perhaps how to interact better with them in other eco-evolutionary studies. Okay. Thanks. Any questions?
Thanks so much for that. Does anyone have any questions? You can unmute yourself or you can type a question into the chat and I will read it out. I guess I have a very basic question, which is, what's next? Like, what's the next step in improving this methodology? Or do you think this is it? It's done?
So, I mean, I think that this is a good first step. And I think that the important thing is that we should be thinking of generalist relationships in terms of beta diversity. Like we should be really trying to account for this spatial heterogeneity that we think is like a fundamental feature of generalist relationships that are really important when we're trying to classify them and categorize them among, other things who we also think are also generalists. And so I think for the method, the calculation itself, I think, is a really good start, and I think that it's great. I think that the next steps and the most important things for your system and for my system as well is just to gather more data. So in order to really have a good understanding of how these things are changing over space, of how these relationships are changing over space and time, we need to actually be out there gathering the data to know what those relationships are. So, for example, the orchid pollinator data set that we used in the paper was gathered over many years and very diligently gathered by our collaborators, Benny and Gary. But of course, like you have to think that a lot of these pollinator observations are occurring at one site, at one location for one time for these orchids who we know have very, spatially variable associations. And so I think the next step for this, like if we want to use it and we want to use it well for interspecies interaction, is to be able to have and to conduct more in-depth and broad-scale ecological studies of these relationships across the ranges of our focal species, not just at one site.
Cool. Great. Yeah, no, that sounds like a really sensible thing to move with this next. So the question from Alberto here, who says, can the package calculate T for focal species only without the matrix of interactive species?
I could write a function for that. It's pretty easy. So it's basically just so Andres Baselga has a great package already for calculating beta diversity for things, and T is just the formula of Simpson of turnover minus nestedness divided by total beta diversity. And so if you wanted to calculate beta diversity like for a specific species, but I like that's pretty easy. I could write that function. So yes, later in like a few hours, I guess.
Anybody have any questions, wants to unmute themselves and shout a question out, or anybody want to type one into the box? I'm being quiet in case people are taking ages to type. Please do feel free to unmute yourself and ask a question as well if you'd like to.
Yeah, so the value is just always going to be sorry, yeah.
It's from Pietro asking, how do you assign the value between the focal and the interactor species?
Yeah, so the value is just always going to be 0 or 1, and the value is 1 if those species interact. So if I know that a fly is pollinating my orchid, that has a value of 1. If the fly is not pollinating that orchid, that has a value of 0. And so really what it is just a table of relationships. So of pollinators that I know for my orchid. The way that you get zeros in is when you have many different focal species who have many different relationships that don't always interact with each other. And so you can end up like you might start with a matrix that's like every pollinator that exists for native North American orchids and every native North American orchid. And that would be like a kind of what you might think of as like a traditional community matrix where the columns would be your focal species and the rows would be your interactor species. And when you melt that, so when you put that into a long form data frame, you end up with zeros and ones. And so that's just like the format of the data frame, but a 1 is just when you know that the two things that you care about, species of the two groups that you care about, have the relationship that you're trying to talk about. So it could be it's whatever you want. Like if you want only things that are known pollinators, if you want things that are known pollinators and visitors, they would have a 1. If you want things that like are known parasites, you would have a 1. If you want things that are no, so just however you're defining the existence of a relationship in your interspecies system is when that relationship exists, it's a 1, and when that relationship doesn't exist, it's a 0.
Cool, thanks. Anybody else? So the next question is from Daniel. It says, in the context of global change, how should we be thinking about turnover importance as potential indicators of disruption to species relationships?
So I think that we can think about it in a couple of ways. So of course, all of these species that we're looking at are specialists and generalists, right? Are mostly generalists. Like we care about understanding this generalism continuum. And so turnover importance is telling us something about species relationships. And I think when turnover importance is high, so when there's a lot of compositional variation in your interspecies relationships, I think that that can tell you two things. I think that it tells you something about the history of that species. So that species has had to have undergone probably a really volatile and really diverse and perhaps like very heterogeneous evolutionary and adaptive landscape in the past to end up having such a variety of pollinators. Or there was some disruption where all 4 of the pollinators existed across that orchid's range something happened to two of those pollinators. And so I think anytime that you have turnover, anytime that you have high turnover in a relationship, that's an indicator of some kind of disruption or some kind of migration or some kind of requirement for the need of flexibility. And so I think that we can also relate that to why we think generalism exists as well. Like, things are generalists survive better and they do really well in areas that are more volatile than specialists because they're more capable of kind of, taking on this change that occurs. And so I think that turnover, like high turnover, is indicative of having taken on some change to some extent. But I also think that having high turnover potentially as a generalist might mean that you're more resilient to disruptions to species relationships in the future. But that also might not be the case if there are super strong barriers to gene flow between, distinct compositional units, and you end up just completely losing like a whole part of your population when those relationships disappear. And so I think it's kind of difficult to say, but yeah, I think that just like turnover in general is an indicator that those species have experienced change in the past.
Cool. Well, thank you so much, Lydia. This was really fascinating. It's a great presentation, and it's always great, I think, to see these papers kind of presented. It's just a very different experience to reading them.
Yeah.
Yeah, no, excellent. So, sorry. Yeah, so I think we can finish there. I think if anybody has any more questions, I'm sure Lydia would love to talk to you over email. We'll pop this up on the Methods YouTube channel so people can watch it back, or you can refer other friends and colleagues to it if they missed out today. And if you've published a paper with us and you're interested in giving one of these, please do just drop me an email. We're really excited to have more of these because we think they're really great. So thanks again, Lydia. Thanks everyone for coming. And yeah, we'll see you next time, hopefully.